All right, so welcome back. This is Nurse Franny. Thank you very much for joining again. This is episode, I think it's 5 or something. We're going to continue talking about genetics. We're not really done. Now, on the previous episode, we were looking at the law of complementary base pairing. We learned what a base triplet was. We learned what a codon was, anticodon, DNA polymerase, mRNA, messenger RNA with its transcription process, transfer RNA with its translation process. We saw what a peptide bond was, amino acids. There's 20. 12 of them can be synthesized by the body. However, 8 of them are essential, which means that we have to consume them. We have to eat to consume these. We need to know what now actually, we're going to learn what a gene is. The current definition of a gene is an information-containing segment of DNA that codes for the production of a molecule of RNA that plays a role in synthesizing one or more proteins. Okay, so a gene is information in the DNA, right? There's a segment of your DNA with information that codes for the production of an RNA that plays a role in synthesizing one or more proteins, and that's what we saw in the drawing. Okay, that's what a gene is. We also need to know what genome is. I'll let you find that out. We need to know what chromatin is, and I'm going to go ahead and draw and see what that. I'm going to try to see what that looks like. We need to know genotype. We need to know phenotype. We need to know karyotype. We also need to know what a chaperone protein is. Chaperone protein. We also need to know, the cell cycle. Cell cycle is very important. So let's see. For the cell cycle, we have obviously interphase, and we have mitosis. And then mitosis, I'm going to teach you the mnemonic PMAT. Okay, for prophase, we have metaphase, anaphase, and telophase. And we're going to learn about cytokinesis. And I'm just going to cut it right there for now, because I'm trying to be wary of your time. So let's start with chromatin. What the hell does chromatin look like? Chromatin is untangled. It's untangled DNA. All right, it's not in a helix. It's just untangled like this. If we zoom out, can't really do that. But this right here contains all of the information that we need, right, for the DNA to then code for RNA to then create one amino acid to then create a protein. Okay, I know that doesn't look pretty, but chromatin, you see it right here, this little mess over here that I made right here, that's just untangled DNA. Okay, so that's that. Let's create a new sketch. So a genotype and a phenotype and a karyotype. Karyotype, we can say it's a chart of all of your, of your 46 individual chromosomes, right? It's a chart of an individual's chromosomes. The phenotype is actually the visual representation of a genotype. So your genotype is actually a particular trait, right? It's the information of a particular trait. However, the phenotype is a visual. So you've seen someone with blue eyes. The blue eyes is a phenotype of that person. But the genotype is a gene that actually codes for a phenotype. Karyotype is just a chart. Your chaperone protein is an old protein that picks up a new protein and it helps it actually fold and bend in its tertiary or secondary structure. So the primary structure is simply the sequence of amino acids of a protein, and the secondary structure is the alpha helix or the beta fold. However, a chaperone protein is an older protein whose function is to assist new ones into bending in the proper shape. We have the cell cycle. So this one I'm going to try to depict it as best as I can. So I'm going to draw, let's see. So here we have G1. So G1 is your regular growth, regular time, right, for the cell. The cell is doing its cell thing here. Everything's good. It's functioning normal. Now S for synthesis. Super misspelled that. S for synthesis. This is actually when the cell begins. So I'm going to call this 1. So here, this is where the cell begins to duplicate. It's DNA. Okay? So the cell, and we have to absolutely understand this, here, so right here, the cell has 46 chromosomes. Okay? Very important to know that, because here, since the cell went ahead and duplicated, I'm making a mess here, but since the cell duplicated its DNA, it now actually has 92 chromosomes in this phase. So when it's going from synthesis, or the S phase, all the way to G2, it brings along 92 chromosomes. And G2 phase, I like to call it the prep phase. The prep phase, because this is where the cell is saying, okay, well, I have 92 chromosomes, right? Now I need double the amount of mitochondria. I need double the amount of endoplasmic reticulums. I need, double the amount of lysosomes. I need double the amount of every single organelle. And once the cell understands that it has enough space, it has enough nutrients, the growth factor, then it begins the mitosis. So all of this, these 3 steps are called interphase. Forgot a 3 here. And this 4th step is called mitosis. So if you were to divide the cell cycle in 2, it's mitosis, where the actual cell begins to divide, and interphase, which is 1, 2, and 3. So you have G1, you have S, and you have G2. Now for mitosis, I said that we have the mnemonic PMAT. So prophase, metaphase, anaphase, and telophase. Here, here, and here. For the sake of condensation and to actually summarize this as much as I can, in prophase, your chromatin condenses. So I'm going to put here condensed into what we know as a chromosomes. In the metaphase, interestingly enough, they actually line up in the equator. So line up in equator. In anaphase, we have your organelles and your chromosomes they actually move to their poles, right? They actually move each to one side of the cell, and in telophase, we have the final divide. I know this is an extreme condensation of what happens with a cell, but for the sake of the exam, for you to actually identify what happens, for you not to get confused, you have your prophase, which is condensation of the chromatin. Metaphase, they line up in the equator. Anaphase, the organelles and the chromosomes each go to one of the poles. And telophase, you have that final divide. Now, cytokinesis, which starts in anaphase, and I'm just going to color these two corners here. Cytokinesis is the actual pinching of the cell. You know, it's the actual moving, right? It's the actual physical divide of the cell, and it looks something like this. So if this is your cell right here, when you see this little divot in your microscope, you see this little indentation, and this is where the cell actually wants to split away. You have your nucleus here. Well, not really, but you don't have that there. But once this happens, this is your anaphase. Once this begins to happen, and it begins in the anaphase, this is called cytokinesis. And at the end, when the cell finally divides, when the cell is finally goes from 1 to 2, you have your two daughter cells. That is telophase. So we went over interphase and mitosis, PMAT. We know what prophase is, metaphase, ana, telo,